this question's from Mani Kanta. What are the challenges in the development of high concentration formulation for mAbs/biosimilars and potential solutions?
Mm-hmm.
Challenges in
What
the
are the
development of high
challenges?
concentration formulation. Yep.
High concentration formulation solution. So I think the high concentration formulation of product in the drug substance in the final solution. I think there are a lot of questions around the stability of the final product and the format of applying it, and on the DP side, right? That is those are the two subjects that come to my mind, right? I think when you look at basically formulation development part, and I've often seen, for example, ranges of, let's say, 50 to 100 milligram per milliliter concentrations. And but I've also seen actually a very high concentration of them. So it obviously depends on the type of products and the mode of administration. A medicine that is actually being injected on the brain, right? It has a very different concentration that the one that is injected in the veins. So, so therefore, when I look back and at the issues that we faced with, were more those shelf life stability studies, accelerated stability studies, freestyle studies that were needed to perform to demonstrate matching shelf life comparable to what we have for a classic mAbs. And there we didn't want to actually end up using newer type of excipients. We often actually, what I have seen is that companies tend to fall back on a standard component library of 20-25 components for their formulation parts. And after ensuring that, yes, this is the final product, then they tend to actually make it more science fiction to actually use components that are less common. And I think one of the challenges there is obviously the supply. And to be sure that those raw mats are actually, those excipients, you're sure to supply them at GMP grade and at the right amount, right? And that is not something that often in early development people immediately think of, right? But I've seen examples, especially we had in COVID time, that there were components that there are 4 companies selling them, but they are all 4 are actually buying from one company, and that company had FDA warning letter. So it was very hard in a way in that time to ask to see that, right? But I think there's another question actually, why and what is the trend with regards to use of those high concentrations, right? Because it obviously comes with certain complications there.
Yeah, yeah. Amar would like to know, actually, before we get to that one, Mani Kanta actually had a good related question that I want to get to. As we move towards higher titers and upstream cell culture, how can downstream handle higher level of impurities efficiently? So, you know, for every action, there's a reaction. How does downstream handle higher level of impurities efficiently when you're getting higher titers upstream?
Yeah, so I mean, there are several strategies that I've seen that people apply, right? One is actually to handle with impurities before actually getting them introduced to the downstream. Techniques like any method of precipitation, flocculation. In those ways, you're actually able to immediately purify those components. 2 is to avoid propagation of extra impurities in your culture by having more efficient, intelligent harvesting, right? So if you do not do that, then you actually release cell debris, LDH, and everything in your culture, and then you contribute to those steps. Third is actually when you have more products. Imagine, for example, you're producing 3 kilogram or 4 kilogram of antibody, and then suddenly you have a process that you generate 8 kilogram DS at the end, right? This is crazy because no one has actually experienced with it in your facility. So I remember actually, I don't know, many years ago in my previous facility, we did actually a thorough debottlenecking of basically, are we going to Tetris is that game. So you have a
Tetris, it
facility.
just came to me.
Yes. So we ended up actually playing Tetris in that area because you have these large containers and you need to move them around and then squeeze between them. And if
Yeah.
someone's foot goes on one of those tubes, you have your water filtration error, right? So the debottlenecking, I mean, it's easily, I was responsible for 10-year CapEx plan for my facility. So part of those questions was actually, well, guys, we come from a 0.5 gram per liter titer 10, 15, 20 years ago, and we are at 4 to 5 gram per liter now. And it is feasible to consider that we actually go to closer to higher end of 5 to 10 in the coming 10, 15 years. So how are we actually getting ourselves ready for that? The fifth approach is actually the consideration for intensifications. There are a lot of brilliant technologies out there that when tested and fit for your molecule, right, they can significantly reduce the footprint, the processing time, and they reduce the impact on your. They have an impact on your end-to-end operations. So that is also the intensification and high throughput assessments that actually can be performed there. I think these are some of the points that I've seen around me that people utilize, right, to address the large amount of antibodies. Yeah.